In the last few decades, a revolution has swept through biology. Researchers now have the tools to unravel the greatest riddle of all, how one species turns into another. The first clue came in 1953 with the discovery of the structure of DNA. DNA carries the genetic instructions for building life. It's passed down from one generation to the next. Unravel its coiled double helix structure, and you find stretches of code called genes. We have around 22,000 genes in our DNA. They carry the information that determines eye color, height, and just about everything that makes us, us. It's the genes that tell an embryo what to become. One set of genes will produce a fish. Another set will make a frog. And it's the genes that tell scientists how one living thing transforms over millions of years into another. At the University of California in Berkeley, Professor Mike Levine and grad student Brad Davidson have discovered that changing just one gene can make a huge leap in body design. They study sea squirts.
Well, it looks humble, doesn't look like much when you compare it to most other animals, but in the laboratory, it's a star.
Researchers consider the sea squirt larva a good approximation of what our early human ancestors might have looked like around 550 million years ago. If they're right, then the human heart evolved from a heart like the sea squirts.
Our heart has 4 chambers. Frogs or snakes, their hearts have 3 chambers. And then you go down to fish, they have 2 chambers. Well, the sea squirt heart has only 1 chamber, but it is unquestionably related to our heart.
So can this humble creature tell us how our heart evolved? At a harbor near San Francisco, the team collects sea squirts for an experiment. Back in the laboratory, Davidson extracts and fertilizes their eggs. He then zaps them with an electric current to insert a loop of DNA into the developing embryo. The operation is equivalent to changing the activity of one gene in the squirt's DNA, but it has a remarkable effect. Under the laser microscope, Levine and Davidson watch the cells that will form the one-chambered heart. An extraordinary transformation takes place.
If you look at the sea squirt embryo, it has spare parts. It has extra cells. Normally, those cells form tail muscles. And what we did was to convert those tail muscle cells into extra heart cells.
Instead of a single chamber, the sea squirt's heart now has 2.
And in this case, there's a nicely formed long heart here. And then there's this little extra chamber up here. So you'll see these individual blood cells making this kind of S-like twisting motion, moving first through the long chamber and then up and back through the second chamber.
We're not just getting a heart that looks like it has 2 chambers. Those 2 chambers are really functioning.
With one tiny genetic change, they have turned a simple heart into a more complex one in just 1 generation. Changing genes in the laboratory is one thing, but for evolution to happen, genes have to change by themselves in nature. It turns out that all animals, including us, have a way of making this happen. To pass the instructions for life from one generation to the next, an animal has to copy its DNA. And whenever anything's copied, errors can occur.
But the enzymes that are responsible for constantly replicating DNA do not do it with 100% precision. They're a little off. It's almost as if there's a built-in mechanism for change.
These random errors are the building blocks of new species. When the DNA changes, genes change, and animals change too. But research in the last few years has thrown up a mystery. When biologists started decoding the DNA of plants and animals in the 1970s, they assumed that the genes of one animal would be very different from another. They were in for a shock. It turns out that when it comes to genes, we humans aren't that different from other species. Around 96% of our DNA is the same as chimpanzees. Around 75% of our genes are the same as dogs. And we share around 1/3 of our genes with daffodils. So if our DNA is so similar, why do Earth's creatures all look so different? To find the answer, we have to look at one of the most amazing transformations that ever occurred. Whales and dolphins are not fish. They're mammals. About 55 million years ago, their ancestors gave up the land and began to live in the sea. At Stanford University, biologist David Kingsley is investigating the mystery of how the whale lost its legs.
If you look at a whale, they're streamlined, they're able to swim at high speeds in the ocean. The streamlining has been achieved by the loss of what would normally be a hind limb structure.
The whale's ancestors transformed their front legs into flippers. But how did they lose their hind legs completely? What change in DNA caused this to happen? The answer to this huge problem comes down to one of the smallest animals on the planet. Sticklebacks come in two forms. Some have a hind limb, and for a good reason. In this rare film, a trout attempts to swallow a stickleback, but the hind limb makes the stickleback too wide to swallow, and it escapes. But in ponds without trout, some sticklebacks have lost their hind limbs, and this provides a clue to how the whale lost its legs. In his laboratory, Kingsley and his team looked for the genes responsible for the stickleback's disappearing limbs. They found that the development of the fish's hind limbs is largely controlled by a gene called PIDX1.
It's only a single gene itself, but it's a white-collar gene. The way that a hind fin develops requires this manager gene turning on very early in development and coordinating the activity of a whole series of other genes.
Fish with hind limbs and fish without them both contain the PIDX1 gene. What makes them different is not the gene itself, but a nearby piece of DNA. In sticklebacks with hind limbs, a piece of DNA called a regulating switch responds to a chemical stimulus and turns the PIDX gene on. The hind limb grows. But in sticklebacks without hind limbs, a small change of DNA in the switch has inactivated it. The gene doesn't turn on when it should, and the hind limb doesn't develop. Kingsley and his team now think that what happened in the stickleback also occurred in whales. The discovery of switches that turn genes on and off reveals how one species transforms itself into another. It also explains why the genes of animals are so similar. The new science of genetics shows that evolution can occur faster and more easily than scientists thought. For over 140 years, people have attacked Darwin's theory. But look at the evidence, and the objections don't stand up. One by one, science has proved them wrong. The evidence is so overwhelming that knowledgeable experts now regard the theory of evolution as fact.
Was Darwin wrong? You're serious. Was Darwin wrong? He was one of the first people with the intelligence and imagination to get it right.
All of this information, this vast crush of knowledge, has vindicated Darwin in